Welcome, everyone. I am Soumya Vemuri. Thank you for joining today's session, using better visualizations to elevate your data's insights. Please welcome our amazing speaker, Mansi Aggarwal. Mansi is a data scientist and an advocate for equality in STEM. Having previously worked for 3 years in various data and machine learning roles within the IT and financial industries, she is currently pursuing a master's degree in data science at Monash University, where she is preparing to undertake the research pathway with a focus on bioinformatics and melanoma research. She is interested in data storytelling and helping business teams better define problems by communicating clearly and effectively with both technical and non-technical audiences. She's also a leadership fellow for the data science track at Women Who Code. Thank you, Mansi, for being here today. I will now pass the session over to you.
Thank you, Soumya, for a great introduction. Hi, everyone. Good morning, good evening, good afternoon, wherever you're joining us from. So let's start. I am going to talk about how you can use better visualizations to elevate your data insights. Little about me. My name is Mansi. I am the 2022-23 fellow at WWCode Data Science. I am working as a data science intern at LongView, a real estate company in Melbourne. I'm a master's student of data science at Monash University. I'm interested in bioinformatics and started working on my master's thesis project in melanoma research this semester. And when I'm not working or studying, I am an avid sitcom enthusiast. So let's get started with today's session. We'll quickly go over the agenda for today. We'll talk about why we need visualizations. We'll talk about what are the pillars of good visual storytelling. I'll show you how to improve very basic scatter plot, and then we'll go to a very interactive exercise where we'll see how we derive deeper insights using visualizations. So, why do we need visualization? We need visualization for enhanced comprehension. Sometimes raw data is not comprehensive enough or does not offer any insights. So we try to visualize the data to uncover patterns and trends or effectively communicate our messages. Sometimes, like I can see a very long time series and not know where the sudden peaks or drops are. So sometimes visualizations help us detect anomalies and outliers as well. It also enables improved decision making and supports storytelling because sometimes there are people who might not be from a technical background or might not understand your technical jargon or domain jargon. So, visualization helps those people understand your data and what your uh, analysis wants to convey. What are the pillars of good visual storytelling? So, the foundation of a good visual storytelling is the purpose. Why am I creating this visualization? Who is it for? What do they need to understand? What actions do I need to enable? And what is the most important takeaway message? If all these things are clear, I lay a very strong foundation for my graphs or charts. Then comes the content, which is informed by purpose. So I need to decide which data to put or include and to exclude. A lot of data creates confusion or creates misdirection or maybe sometimes confuses the audience. So it's very crucial that you decide which data is absolutely necessary to put into your graphs. Then comes the structure. So structure also involves which kind of chart or graph you want to use. For example, you use bars for comparison, line charts for time and continuity, stack bar charts and pie charts for composition, although personally I'm not a very big fan of pie charts, scatter plots for correlation, and heat maps for volume of location and events. And finally, formatting. This helps us highlighting what's important. This improves aesthetics, adds appeal and focus, and it's just like icing on a cake. So, as I discussed, raw data hardly conveys useful information. So, I can see a table, which are very important. Most of our data exists in tabular format, or we try to bring it to a tabular format so that it's more shareable and accessible. But what is MH and DH in this table? I'm not sure about it. I might have metadata, but what if there are 20 columns or 200 columns? I can't go around carrying my metadata and like explaining each and every column like that. So good visualizations make this information accessible. So once I've plotted this two variable table, we now know that this data has mother's height and daughter's height. And this also tells us that there is a strong positive relationship between the two. So, like I discussed in the content or structure section, that we also use different type of charts to show different types of information. So here I might want to focus on the relationship between the two variables. Hence, I use a scatter plot. So, how do I go about improving the standard chart? Less is more. We always start simple and then work our way up. And another method is using clear labels and subtitles, titles and subtitles. So your title should be succinct with information. It should be short and descriptive. Subtitles adds more layer of information, which if someone just wants to glance at the title, it's basic and necessary. Subtitles just adds more information. Customize your charts, use non-default colors and settings. Try to emphasize on key information. Test for accessibility. This is very important. Sometimes when you're like, everyone works online now, so we have to make sure that whatever we are presenting is accessible in terms of the colors that we're using, that they go together. Sometimes the two colors that we're using are juxtaposing each other and we can't see what is important, and this might fail, like this defeats the purpose of a visualization. Very importantly, seek feedback. Always maybe show your analysis to a person that has not worked on it with you, so that they're able to tell that are they actually deciphering the same kind of information that you're trying to convey. If they're able to do that, then I think you've conveyed that information in a very good visualization. Let's go about like making this chart a little better. So I use a non-default color. I used a good theme. The generic ggplot theme is a basic gray one. I've used a white simple theme. Then emphasizing key information. I wanted to show a strong positive relationship. I can just fit a simple linear model and plot it. Then I go about sprucing it up. So I used a subtitle to show that the outliers are in red. So I used a bright red color to show the outliers. I started small and then I worked my way up. So to begin with, the black and white chart might have been good enough if I had just 1 hour to put up a presentation. So I had a backup plan. But if I have more time, I can tinker with my baseline charts. What I've added here is that I've annotated these outliers with the XY coordinates, and maybe I've like tried to amp it up. Now that we have like gone through the process of like seeing how to improve a standard chart, we might be interested in the main key element of today's presentation is how do we derive deeper insights using visualizations. So for this, we're going to look at an example from the world of bioinformatics. So just a heads up that you do not have to be an expert to follow this. Just see this from a lens of how different visualizations can offer meaningful insights, which might otherwise be hard to decipher from large tables. So what exactly will we be looking at? So I created a report last semester which tried to provide a review of an existing paper where they actually tried to understand the relationship between COVID-19 cells, the virus cells, and the human host cells, so that they can develop some new effective therapeutic strategies. So I had the data, I had the existing research. The examiner wanted to see how I am able to use that information to derive my own insights. So, what are the things that we need to remember as we move forward? You do not have to focus on understanding the meaning of the biological terms. Take everything at the face value. For instance, up means going up, that is increase. If I say down regulation, just figure out something like just imagine something going down. Significant means important, and not significant means not exactly not important, but like if you are statistic, like if you have statistics knowledge, so here significant actually means a p-value of less than 0.05. So just focus on these words in English or statistics. We don't have to understand the biological terms. What is our data set consisting? So we monitored the host cell response to infection at various periods. This is important. We have 4 time periods here: 2 hours, 6 hours, 10 hours, 24 hours. And following the infection, and studied 2,715 significant proteins. So this was my starting point, a very average chart. I just plotted all the proteins I had with their p-value on the y-axis and the ratio at the 2 hour time point on the x-axis. Don't have to learn what ratio is. They're just two variables, and I've plotted their values for all the proteins. What is going on? I did not know either. This was my baseline chart. But now we'll see the journey of from where I went and what analysis I was able to produce only through visualizations. We clearly need to amp this up. Let's focus on what information do we have. We had information called ratio. So negative means down, positive means up. P-value, this is a metric for statistical significance, that a p-value of less than 0.05 is important to me. Gene symbol. So this allows for clear and ambiguous reference to genes. There is a nomenclature. For example, CO2 means carbon dioxide. This is a set nomenclature for that element. All the proteins and genes similarly have symbols. We have to use information to our advantage so that we can create better storytelling. What I did to that standard chart is in front of you. We just translated all information in the form of visualization so that I can able to see all the information very clearly on this chart. What I did was I created a line to set a threshold of 0.05 set for the p-value. So above this is significant and below this is not significant. Then I set a threshold for the ratio, and I created 2 vertical lines. That means I know that a positive ratio means upregulation, but I want to see the genes with a ratio above 0.5 and below negative 0.5. That means these are significantly upregulated or downregulated. Now I got my target area, which is these two squares, these two areas that are significant also and significantly up and down regulated. What I've done is I've just taken these 10 top 10 proteins and I've mapped them here. I hope you're all following me. Soumya, just give me a heads up if there's any issue anywhere. What I did was I performed all these 3 steps for all time points: 2 hours, 6 hours, 10 hours, and 24 hours. When we did this, we found that the significant proteins, the 10 that I plotted, are different across all the time points. This is something that I was not expecting. I thought that the important proteins will carry out the journey. I'll see a pattern throughout the time points. But this is also important to me. That means that when the disease progresses, the the reaction of the host changes. The initial onset of the disease is later than the progression of the disease. What I did for my project was I wanted to investigate the significant protein at this time point to understand the later stages of infection and how the body reacts when the disease is a little stronger in the host cells. Now I want to bring your attention to a important topic, that is that same information can be conveyed in different formats. So what I did was I took these 10 proteins and I mapped these 10 proteins on the previous time points. So I'm like backtracking my information to see how these 10 proteins behaved previously. So I just took the 10 proteins. Calculated their, subsetted their ratio at 2, 6, 10, and 24 hours, and just drew a line plot. What I can see very clearly here is that 3 proteins are showing an upward trend, but all 7, the majority, are showing a downward trend. It might be worthwhile in my later stages of the study that I might, analyze these 7 and these 3 proteins a little differently. But this only shows me the trend. What if I want to show the magnitude of this change? I use a heat map. This conveys a similar information that this is going from light to very light. That means it's going down. But these 3 are going to a darker shade. That means upward trend. So it depends on what information you want to convey, the magnitude of change or just the change, the pattern of change. So I hope that all the 4 steps, all the 4 pillars are covered here, and that you understood how to go from a very basic chart that I plotted all the 2000 proteins, and then like slowly and slowly I amped up my visualizations, used all my information to my advantage, and was able to generate these insights. So in conclusion, purpose is very important. You need to have a clear idea of what you need to convey and present. And sometimes what happens is you go reverse engineer your procedure. So first I need to plot all information and then build up and define my purpose. So in the middle stage of the analysis, I define my purpose that I now want to consider the later stages of infection, and now I'll explore the proteins significant at 24-hour time point. So this is something that you have to be very clear of what you need to convey and present. Always start small and then increase your complexity, and always have a backup plan. I personally use RStudio and the library called ggplot for my visualization tool, and you should also focus on mastering one visualization tool and then dabble with more. Sometimes I use Seaborn and Matplotlib libraries in Python. I've personally not used Tableau or Power BI, but I've seen results which have been amazing. But I think you should pick one tool that comes naturally to you and then focus on that first. Experiment with colors, annotations, chart types. Maybe you would want to have specific custom fonts for yourself. Maybe download fonts. There are libraries to set fonts for your visualizations. Maybe you want to have a palette that you want to try, and you can create your own palettes with a set of colors that you want to have in your visualizations. Or you can have a branding guideline which uses colors from your own brand and logos. So experiment with all this. And last but not the least, this is very important, practice. Practice making complex and interactive visualizations when you have time. You will not be able to create very high quality graphics in your first try. It is a process, it's a long process, and you have to practice with a tool of your choice. So spend time with it. Uh, Obviously, not when you have a deadline, but spend time with it when you have time. So some of my favorite resources that I have learned from over the past years. First is the Tidy Tuesday project. This is a weekly social data project. They release the data every Tuesday. Everyone is welcome to participate. You can use it for statistical analysis, building machine learning models, and maybe practice your visualization skills. An excellent repository for chart types called From Data to Wiz. This is based on the input format, and it comes in the form of decision trees, leading to a set of potentially appropriate visualizations to represent your data set. A very, very, very engaging tutorial on ggplot, beautiful plotting in R by Cedric Scherer. It's an extensive tutorial, and it gives a lot of examples and step-by-step process on how to modify a ggplot. So I showed you the journey. This tutorial will tell you exactly what steps you want to take to follow this journey. And another community-driven project is the 30-day chart challenge, which happens in April. It's an event with the goal to create a data visualization on a certain topic for 30 days in the month of April. So you can follow all these resources, and I hope you learn a lot from that. That's it from my side. Thank you, everyone. Thank you for listening to me. Thank you for giving me your time and this platform. And thank you to Women Who Code for lending us this opportunity to put up all these great talks for everyone and all our community members. This QR code will take you to my LinkedIn. You can connect with me, drop in, say hi, show me your visualizations, or just if you have any questions about my talk, if you want these slides, I can send them to you. So thank you, everyone.
Thank you, Mansi, for presenting on how we can use better visualizations to elevate our data insights. That was an amazing talk, and we all learned so much. Um, Everyone, please feel free to share any questions that you have for Mansi in the chat. While everyone is sharing their questions, Mansi, would you like to share a little bit on what kind of career opportunities there are for someone who learns or who gains an expertise in creating visualizations or getting better insights in data with the help of visualizations?
Oh yeah, so definitely UI and UX, graphic design, great career options, and I've seen great freelancers working on this. If you connect with me on LinkedIn, I can like share those profiles with you, anyone looking for that. And Cedric Scherer, which the repository has shared, he's an excellent person. Like I have followed him for quite a few years now. I think he's a great person to follow, and he actually, like does visualization full time, and he makes amazing interactive visualizations. So there are great freelancing opportunities in visualization.
Awesome. So we have a few questions coming in. This one's from Karen. Do you have a standard process for visualizations, like a list of visualizations you always apply on the data on hand?
So, like my recent work for the past few months has involved a lot of correlation analysis. So, obviously, scatter plots. I have my own code repositories so that I don't have to like do the color palette setting, and like I have like code templates that I can use directly. But I have found myself using line charts and scatter plots a lot. But again, it depends on your data, and as I showed that I actually created the heat map for that trend because my examiner wanted to see the magnitude of change as well. So it also depends on the data and what you want to convey or what you are required to present.
Yeah, there is a lot we learn along the way as well. So, like another time, we would start going into the heat maps. Another question from Annie. Mansi, quick question. What advice would you give to someone who is struggling to identify what graphs to create from a complex data set?
So again, first of all, it definitely comes with practice. For like usually, I used to struggle 3 years back when I used to be given handed out a data set, but over the time, I think when you explore different resources, you find that you're following a specific kind, like more and more, and I think then you learn how to use a resource properly also. So the resource that I shared from data to Wiz, it actually guides you that if your input data is in this form, what decision tree you could follow to actually reach a visualization that is suitable for your data. So that might be worth exploring. I actually use that sometimes myself.
That's awesome advice. Wendy Marissa has another question. Are there any guidelines to visualize data for visually impaired audiences?
I am not very sure that this means like entirely visually impaired. I'm very sure that accessibility guides or alternate text that exists for visually impaired audiences. But if like people with color blindness or any other issues identifying certain colors, I think, for example, Women Who Code, for when I started as a fellow, we had an accessibility guide, which color goes on which and which shouldn't be used with which. But I'm sure there might be, if one Google search away, there might be accessibility guidelines for the usage of colors and how to represent them uh, well. I mean, there must be data for that as well.
Yeah, that was a great question. Okay, we have one. Um, what advice would you give to a sophomore majoring in data science? Would you recommend joining a lab or looking for an internship?
So this is like question is twofold. It also depends on an interest. Do you want to go into research, join a lab, or do you want hands-on industry experience, look for internship? Personally, both my undergrad and postgrad universities had opportunities on campus where they would tie us up with industry partners. I'm sure looking up that might be worthwhile. Second, always use look for external resources as well. Like for example, there are boot camps which like help you do a capstone project and end. And I'm sure like Women Who Code has hands-on series as well, where you can actually build a project with us or like do capstone projects with like after doing a series of tutorials. And you can definitely reach out to me after the session, and maybe I could personalize this advice a little more.
Awesome. Do you think a degree in AI or ML is necessary to move into the field?
I have seen many self-paced learners as well because there are a lot of online resources available. And but if you are going for the self-paced approach, it is very important to apply your learning. Like you have to like figure out how to apply that learning in the form of doing your own project, or like I said, like uh, joining a team, or like going for doing a kaggle competition, or like taking up volunteer roles where you can actually apply your learning. And a degree in ML or AI is like a point of privilege also, because it requires investment, taking a break from working. So if you have that privilege, you can go ahead for that. Personally, I wanted to do this degree because I wanted to explore the field of bioinformatics, and that is something which was not coming to me via work experience because I don't have any prior biology knowledge. So my university had a research stream specifically for bioinformatics, and if you apply and qualify for that, you can take up that. So yeah.
Awesome. Uh, everyone's loving the talk and a lot of insights that you shared as well. They
Thank
found the
you.
presentation very helpful, and everyone's very grateful for the talk that we had. Thank you, everyone, for sharing your amazing questions. I want to give a big
Thank
thank you to
you.
Mansi again for
Thank you,
the
Soumya.
time that you spent and the amazing talk. I want to thank everyone so much for joining this session and for being a spectacular audience.